Hi everyone, good afternoon. So I'm Gabriela, I'm the General Coordination of XXI, and today we're starting our second international webinar. I'm glad to have everyone here, and just remembering the attemptee list will be on the chat in the end of each lecture, and you lay your questions in the chat too, and we'll pass to the presentation. Thank you.
Hey everyone, my name is Kai. I'm also in the coordination. So I want to extend our gratitude to all of the sponsors who made this event a success. So our biggest gratitude goes to the gold sponsors like Wizard and Aeroclub Labras. And we'd also like to acknowledge the support of our silver sponsor, Say Yes. So it's a really important to us that you believe in our vision. So thank you for all the sponsors for the support, and we sincerely appreciate all of your contributions to this event. Thank you so much.
Our first lecture will be presented by Arthur. He's research coordinating in GDM. He works with diploids in maize. So I'm gonna share my screen with you, and then you can see the lecture.
Can you hear me now? Okay. Yeah, it was muted. Sorry. My apologies. My bad. Sorry. Can you guys hear me now? Okay, let me share. Yep, let me share my screen here. Let me know if you can see my presentation, okay? Okay, can you see? Okay. Okay, good evening, everyone. I don't know how many people we have here. talk. And I will try to be short and explain a little bit about my job, about what I do, and what I believe as a scientist that will have a huge impact in our food production system if we really want to do that in a sustainable way. Okay, so the title for the presentation is "Shaping the Ag Future." So the ag, it's a short version of agriculture for those that don't know what is the meaning of the term. And the subtitle, I wrote this on purpose, okay? Because um, nowadays, we as a scientist, and I believe that many people here in this room are scientists too, or are being prepared to be someday. And when I say beyond excellence, is because to be great and to be good, it's no longer enough. Okay, so we must go beyond the excellence if we really want to strive as a scientist, as a scientific community, especially in plant breeding. Okay, so uh, let me see if I can move here. Uh, when we talk about plant breeding in general, there is usually a paradox. It's when I say a paradox, I mean it's a question that for I don't know a century, it's there is a huge discussion around this question: plant breeding, science or art? I don't know how many people here in this room are coming from the plant breeding community, but in plant breeding, we are all the time trying to answer this question. For some people, plant breeding is a science. It's completely science. It's statistics, biology, genetics, plant breeding methods. For some people, plant breeding is like more an art, like the beauty of art, the beauty of making crosses, making visual selections. Some people consider that as an art part of plant breeding. And for some, it's a combination of both. But regardless that, if you believe if plant breeding is a science or an art, we have an equation in plant breeding that drives basically every single decision making process that we as a scientist we take. And this equation is the genetic gain equation. The point here is not to go in deep detail about each factor of the equation, but just to give you a bigger picture. When we talk about genetic gain, we have a few factors that we must take in consideration, and we have intensity selection, we have accuracy, we have genetic variability, when we have time and breeding depression. And the part of my job as a plant breeder, it's I work with double haploids, and later on down the road, you will see how double haploids play a significant role in the genetic gain equation, and how we can leverage double haploids to make, to produce more, to create hybrids, to create inbred lines for the development of a much better food production system. Okay. Here we have three main things here that I want to make a comment about. And when we talk about plant breeding, and we have the line development part, and you as a scientist, as a breeder or part of the system, you can produce your maize inbred line through two different ways. One is conventionally, and the second one is through the use of double haploids. So, what is the difference between both? When we do line development through conventional breeding methods, there is a huge amount of time that we need to take in order to produce the lines. Because before we can make a hybrid, like in our pipeline, we need to produce the parents of that hybrid. And the parent, it's an inbred line. And in order to produce an inbred line, there are, several rounds of self-pollination if you want to do that through conventional methods. Or you can leverage the haploids and produce your inbred line 100% homozygous, 100% pure line in, let's say, 1 year and a half. That's the difference. So you have conventional breeding methods. We can take, you know, 5, 6, 7 years to produce an inbred line, and we can do that through double haploids with 2 years or maybe a lot less. Okay, so that's the difference. And when we use double haploids to produce those inbred lines, we have also the possibility to apply genomic selection for those that really like the idea of applying molecular markers and prediction models in order to produce a corn hybrid. It's very suitable to do that with double haploids. So that's uh, just a few of the benefits of using double haploids. Okay. However, when we use double haploids for hybrid production for line development, on the left, we have a few steps that we need to follow. Okay, so the first step is you must have a haploid inducer. Okay, a haploid inducer is a type. It's a genotype which has the ability to produce haploids. Like in a very basic way. And when you make the crosses with your source population, okay, so you have, let's say, I don't know, 50, 100 populations, different populations in your pipeline, and you want to produce inbred lines out of those populations. So you make the cross with the haploid inducer, and you harvest those seeds. And among those seeds that you harvest, just a very small percentage of seeds will be haploids. And and how can you differentiate the haploid seed from the diploid seed? Just a small portion, like it depends on the inducer that you are using, but something around 10, 12, 13% of the total seeds in the year will be haploids. And you have a way to differentiate. So there is usually the haploids. So the embryo is completely colorless. In the top of the seed, there is a mark. It's an anthocyanin mark. So that's how you can differentiate the haploids from the diploid. And also you have those haploid seeds. So what is a haploid seed? It's a seed which contain only one set of chromosomes, only the maternal sets of chromosomes. So just a copy of the maize, let's say maize DNA, maize genome. And once you have those haploids, you need to use some products, usually some chemicals, in order to duplicate the genome of the haploid seedlings in order to make the plant fertile. Otherwise, if you plant a haploid seeds, so it will grow a normal plant with the female part okay, but the male part, which is the tassel part of the plant, will be sterile. So we will not be able to produce pollen. That's the problem of the haploid. So you cannot self-pollinate, which is the next step of the double haploid pipeline. So you need to apply a chemical for that. And some people use herbicides, gas. Some people use chemical which is called colchicine. It's up to you. You have a variety of options that you can use for genome doubling. And once you duplicate the genome of the haploids, so you will finally be able to self-pollinate that plant. And once you self-pollinate that plant, that seeds that you will harvest, those seeds are the double haploid inbred lines that you have. Usually something around 4 to 5 seeds, the max. It's sometimes you can get more seeds than that, but just consider 5 to 6, 7 seeds at the most cases. And that seed that you harvest, that seed, it's your inbred line, is 100% homozygous, 100% pure line. So, and you just have a few, and you need to do the next step, which is the seed increase. So instead of taking 5 years, maybe more, you can have your embryo line is just in just 1 year and a half. That's amazing. That's the power of double haploids that we have that we can leverage in maize breeding. However, the process has some challenge, right? So as every single plant breeding method in the world, and we start with the haploid inducer. Most of the time, and I would say 99% of the time, the inducers come from outside of Brazil, which means that the haploid inducers comes from the temperate germplasm. It's not tropical. It's not adapted to Brazil, to the areas of Brazil, which means that when you have that kind of inducer here in Brazil, the development it's not good, not good enough. You have some problems, especially with foliar diseases, insects, development issues, pollen production, pollen shedding issues with that inducer. It's a disaster to work with those inducers. We have CIMIT in Mexico, which affirms that they have developed some, let's say, semi-tropical haploid inducers, but everyone that works with haploid inducers in Brazil know that in reality it's a little bit different when we have to work with those haploid inducers, especially here in Brazil. We have some problems with inhibitors. Why inhibitors? Because inhibitors are like some, like let's say, some genes which is present in the tropical germplasm that does not allow the expression of the morphological marker. So when you have some populations which contains a lot of inhibitors, uh, it's nearly impossible to differentiate the haploids from diploid seeds. Some people say, okay, Arthur, we don't need to use morphological markers. We can use oil content, for example. We have many other ways to do that, but when we deal with large scale uh, production, large scale double haploid production, you need a method which it's straightforward to be used. And sometimes some methodologies are not like that. Okay, so keep that in mind. And the doubling, it's one of the most important steps in double haploids production because it's the part where we use the chemical to treat the plants. It's a colchicine. Colchicine is a chemical that we use. It's a very toxic chemical, and it's used to double the number of chromosomes in the haploids, which make the plants fertile in order to produce pollen, so we can finally make the pollinations. And colchicine is a product that we need to purchase outside of Brazil. There is a cost for that. Usually, it's pretty high. The cost is toxic. You cannot dispose of that chemical anywhere that you want. Okay, and the last but not least is the pollination, because when you treat a plant with a chemical like colchicine, the plants are so weak in the development, and the pollen production is so compromised that in order to make the pollination, so if you have seen a normal a normal maize plant in a field with a lot of pollen, the tassel, like in haploid, which was treated with colchicine, it's completely different. Like they produce a really small amount of pollen. Sometimes you need to be very careful in order to make pollination, otherwise you will get zero seeds at the end, and that would be a problem. Okay, so here I have just for you to understand a little bit about how the method of double haploid works in reality. So you have the inducer genotype. So you take the pollen of the inducer, you pollinate the silk of your population. Okay, so and the result kernel, only the female chromosome will be present in the seeds. Okay, so and the colchicine, the chemical that I mentioned earlier, it's what we call a mitotic inhibitor. So the normal process of cell division when you have the cell in the division process. So the chromosomes, they align at the center of the cell, and the spindle fibers, they attach to the centromere region of the chromosome, and they pull the chromosomes apart toward the poles of the cell. However, when you apply colchicine, that spindle fibers are completely destroyed. Like they do not exist anymore. That's what colchicine does. So instead of the chromosomes in the center pulling apart from the center toward the poles, that process of pulling apart does not happen anymore. That's what colchicine does. So you have in the next season, the next generation, a plant that is completely homozygous. That's your D8 line. We say usually some people say double haploid lines. We can use a short version, which we call D8 line. You can use like that if you want. Okay. So as I said, double haploids. So we have the haploids. It's a plant, okay, with a complete set of chromosomes from a single parent, okay, and we can create those haploids. We can do that using the haploid induction in vivo, what we call. So we have in the field, we have the inducer genotype, and you have your population. You make uh, pollinations, crosses, usually in isolation fields. You can do that manually if you want, if you have budget for that, if you have people to do those pollinations, or you can do that in vitro. Okay, you can just use enterculture. You can just take the pollen and try to regenerate a haploid from those pollen grain. You can do that as well. Of course, the challenge we need to emphasize, because regenerating a haploid plant from a pollen grain, it's not a straightforward process. We have a lot of protocols that need to be validated to do this kind of process. And the importance of this, it will speed up the breeding process. Why? Why speed up the breeding process? It's quite simple. So instead of having 7, 8, 9 generation of self-pollination in the field, you will only have 2, maybe 3. So there is a reduction not only in time, because when you have less generation in the field, that means a lot. And bringing back to the sustainable way of producing food, if you have less in the field, it's less paper bag, less plastic bag, it's less pesticide, insecticide, fungicide, less people working in the field. It's a lot of less. You see, that's how we can produce food in a much more sustainable way by leveraging the use of double haploids. Okay. So as I said, by using double haploids, you can significantly accelerate the process of breeding and reducing time at least 50%. That's huge. And there is another point when for us that work with genetics and plant breeding in general, that we are always looking for genetic uniformity. So that will be able to do that with double haploids, because you have a line that is completely homozygous. Okay. Reduce cost. Well, of course, you have less generations in the field, so you have less time, less options to use chemicals, to use resources. It's a really saving in terms of budget. Okay, and for precision, because it will facilitate you to work with some traits, especially if you work with selection, because when you have those double haploid lines, which are completely homozygous and completely homogeneous, your accuracy of selection is much higher. So consequently, the heritability of depending on the trait that you are working with, it's like what you see is the fact, what is the composition of the genetics of that plant. So your selection is much more precise when you have a line that is much more uniform. Okay. And bring it back to the topic of the talk. It's okay. How can double haploids help us to build a much more food production system, which is a much more sustainable. How is that possible? Okay, we can work with crop resilience. Okay, because through the use of double haploids, we can produce lines. Consequently, we produce hybrids, which are much more stress tolerant. So you can make selection at the inbred line level and select those plants which we identify that they have a strong ability to support the lack of water, for example. Not only water, but also the high temperature, for example. So you can develop a crop which is more resilient, much more resilient. Okay, disease resistance, it's also another point where we can use double haploids to produce those hybrids and inbred lines which are tolerant or even resistant to some diseases. And that means if you can create a line and consequently a hybrid which is tolerant to diseases, that means less fungicide. It's less fungicide in the environment. It's less fungicide, insecticide, herbicide in the soil, for example. So that's how we build a very sustainable food production system. And it can, of course, improve yield. That's the point of every single scientist in the plant breeding community, and reduce the chemical use. That's the point. Okay, if you want to build a much more sustainable environment, we need to use less pesticide. And how can we achieve that? By leveraging the use in the development of germplasm. How that's my job at the company, to develop those inbred lines, to recycle the germplasm, and to develop a um, much more high yield lines, and consequently high yield hybrids for the breeders to test outside of the nursery where I work. Okay. Another point of that I would like to mention is, okay, we have, everyone knows double haploids in maize, right? So everyone, it's okay. We know it works. It's good. It's great. Of course, there are some points that where we need to change a little bit. Some parts where we need to improve. Uh, but we need also to expand the crop range. What I mean by that, we need to concentrate efforts in order to develop the double haploid technique to other crops, not only maize. How about soybean? I know that we have double haploids in wheat, sorghum, and okay, but how about other crops? How about rice? Can we leverage the double haploid technology in rice? Wait, that's people that work with science, research, and development. We need to focus our energy in developing a new methodology for other crops too. Okay. In terms of precision breeding, like targeting specific traits, for example, like nutritional content, we that's something that's kind of interesting because we are all the time looking for yield in terms of grain. We want to produce more with less. But okay, it's totally fine. We can produce more with less, but that more that we are producing, how about the nutritional content of that more that we are producing? It's okay. Let's level up the nutritional content or not. We need to keep that in our mind, and of course, as we move in our life, so the world it's constantly changing, and the climate change it's a reality, and we need to be prepared for that. And how we'll be prepared for the climate change, by leveraging the use of science. Science will help us at the end of the day to surpass any kind of challenge that we can face in terms of food production systems. Okay, so let's develop more crops which are more suited for changing climates. That's must be the end goal of every single scientist, especially in this room. Okay, and well, I believe that that's my last slide. And once I'm done here, we can open for questions. So about the double haploids, of course, there is no question about the importance of this technology, especially in a modern genetic improvement. We can use double haploids to produce pure lines, recycling germplasm, which both are very important in every single maize breeding pipeline. Okay, and by optimizing the use of resources, we can produce corn hybrids, which are much more superior to what we have. Um, and that's, of course, will accelerate the breeding process and play a crucial role in the sustainable food production systems that we have. So, if you think about sustainability, there is no way that you can detach the double haploids from it, because double haploid is a technology that it's here to boost the plant breeding and will consequently help us to build a much more sustainable food production system, that's for sure. Okay, well, I don't know how many minutes I still do have. I just prepare a very short presentation just for you to just to give you an idea about what double haploid can do for you, what double haploids can do for the scientific community, and how we can use double haploids to produce a much more sustainable food production system. So that's basically all that I have. This is my LinkedIn. So it's a QR code. If you just scan with your camera, you'll be redirected to my page on LinkedIn. So if you have any question, or if you want to make some kind of a contact or a connection, a professional connection, please just feel free, go ahead. I will be more than happy to accept your networking connection. Okay, but well, that's all that I have. Thank you so much for your time, your patience. Hopefully I was clear enough with my message, and I hope that everyone has enjoyed the talk. Okay, thank you. Mhm. Thank you. Okay, that's a great question. Uh, so we have some equipment at the GDM. We have a biotechnology lab where we can basically do any kind of research with basically all the crops that we work with. So it's not only maize, soybean, wheat, sunflower that we are trying to implement. But yes, we have some equipment not only to see the number of chromosomes, but also to do sequencing. So to evaluate at the DNA level any kind of research that we have.
Thanks. If anyone
Mhm.
has any question,
And maybe just in case you don't have any question right now, you can just send me a message, my email, or contact me through LinkedIn, and of course we can I can answer any question that you might have.
people are saying thank you here.
Yeah, yeah, thank you. It's quite a challenge to give a talk in English. It's not that easy.
For us too, I think people get really shy to talk.
Oh, yeah, yeah, I see. But it's a great opportunity, and congratulations for the organizers
Thank
of
you.
for this event. It's really great, really
Yes,
great.
it's
Yep.
very important, and your
Hmm.
lecture was very important too. I think it's very actually is a actual uh,
Hmm.
thing to speak about. You know, people
Hmm.
don't really know about it, and it's
Uh-huh.
very big to the science.
Yep. Well.
Gabriela asked it again. Do you
Uh-huh.
personally think that improvement is art or science?
Yeah, it's a tricky question, Gabriela. Yeah, yeah. So, well, um, I well, I think that I believe, okay, that plant breeding it's a combination of both. Like we have a lot of we have a lot of science applied to plant breeding, like statistics, genetics, cell biology. But I like the idea of the art part, of going in the field and see like every single, like morphological structure of the plant. Like when you go to a field corn, sorry, a cornfield, and you're making pollination, and you see a lot of bees, like those. I mean, at the end of the day, it's it's a beautiful place to be. So for me, that part of my job, I consider as the art part. So, but my answer for me, Arthur, I have a lot of friends that will disagree. They will say, no, it's a 100% science, Arthur. It's not art. But for me personally, it's a combination. I would say 50-50.
Thank you. Now,
Yep.
Patricia said, how does germplasm recycling work?
Okay. So that's a great question. Yeah. Um, well, when you produce the lines, the Limber lines, okay, let's say, okay, we have my date lines. I make the crosses, line A and line B, and I make the hybrid of, I make the population of that crosses, and I start working my breeding process in that population. But later on down the road, let's say 7, 8, or 9 or 10 generation later, you have exhausted all the genetic variability of that population. So there is no more genetic variability to work with for that particular population. So that's where you need to start over. So the recycling process, okay, let's take that breeding population and cross again with maybe a different source in order to see if we can find new source of variability, of genetic variability. So that's how the recycling works. In a very simple words, of course.
Thanks. Gabriela said she is agree with you. And now
Good.
Maria Laura asked, first
Uh-huh.
thing,
Uh-huh.
thank you for the presentation, and it was very pleasant to watch you.
Mm-hmm.
Wait, okay. Now she asks, does this technology can be associated with speed breeding?
Mm-hmm. Mm-hmm. Yes, absolutely yes. Yes. We can use. Actually, we have some speed breeding projects at GDM with soybean, and of course, sure, we can use double haploids in combination with speed breeding techniques. Right. For example, there is one technique that I'm aware of. Like, okay, we can use the light, like extended period of light in order to induce the plants to grow much more and faster. And we can combine that lightning with double haploids. And yes, that would be a really nice combination. Thank you for your question.
Now Julia asked, you told us about Cochin. I use it to work
Uh-huh.
with APM, and I learned that APM can be less harmful to the environment than Cochin. Do you ever
Hmm.
heard about something about it?
Oh yes, absolutely. Yes, you are completely right. We have some not some, let's say, a few products which are much less toxic than colchicine. Colchicine is very toxic. The problem is that when you work in a large scale, like when you have a lot of plants to be treated, colchicine it's much more suitable for large scale process, and it's very easy to access and to buy. That's the reason why most people use colchicine. But in terms of being toxic, yes, we have herbicides. Even some gases are less toxic than colchicine.
Now Moisa said, can
Mhm.
DH that be used in fruits?
That's a really great question, actually. Well, I never thought about it, to be honest. I believe that maybe, but I cannot say yes or no. right now, but I actually, the shortest answer would be I don't know, but I believe that it's possible. Like we just need to identify which fruits are you talking about and how we can regenerate. Because the problem is the regeneration of the plant. Because, uh, well, I don't know if you can produce like a haploid inducer for that, but we can use enterculture for developing, for taking those haploid cells of that fruit. And once you take the haploid cell of that plant, you need to be able to regenerate that into a plantlet. And then we can apply the chemical to double the number of chromosomes. So we need to develop a protocol, and to optimize the protocol, the regeneration protocol is the key. So I believe that, well, in science, it's very hard to say, okay, this is impossible. Like I think that it's impossible until someone figure that out. That's what I believe.
Thanks.
Thank you. Thank you.
Uh, Now, Isaac asked, which molecular mechanism and genetic factors are key for inducing haploid plants efficiently, and
Okay.
how it can this knowledge enhance precision in
Ah,
crop improvement programs?
okay. Yeah, that's an extraordinary question. That was really great. A very good one. And well, the molecular mechanism of haploid induction has been already characterized. They already identify the QTL regions. So there is one QTL in chromosome 1 and another QTL in chromosome 9. And they found that both chromosomes combined can explain over 65, 68% of the variance for that particular trait. And they have identified two or three genes that are responsible for the haploid induction. So that information is already public. So there is one center of research and development at Iowa State University in Ames, Iowa. It's called the 8 facility. We have Thomas Luberstad and Ursula Caroline Frey. Luberstad was my major advisor in my PhD, and they have identified that those QTLs in those genes. So, and how we can use that. So, also we have that information available. You can use that information to build your own haploid inducer. Remember when I said at the beginning that most of the inducers they come from outside of Brazil. They have a temperate germplasm. But once you have the region, the genes, you can simply develop markers flanking that region, and you can create your own haploid inducer. You can take a tropical germplasm background and develop your own haploid inducer using that information that is publicly available. That was a really great question. Thank you.
Thanks. I think it's that. If anyone has any questions, guys, I think it's that.
Is that okay?
Thank you very much, Arthur. It was really, really great.
Thank you.
Thank you. So,
Yeah,
bye.
thank you so much. So, okay, once again, thank you so much for your time and patience. And if you have any question, please just add me on LinkedIn. I'm here in Londrina, Paraná state. So feel free to stop by whenever you have a chance. It will be my pleasure to receive you here in our company. Okay.
Thank you so much.
Yeah. Thank you.
Bye.
Bye bye.
So, guys, now we're gonna go to our little break and we'll be back soon. So, hey guys, we're back. The attendance list is already in the chat. Stay tuned. And now I'm gonna thank you again, our sponsors, Wizard, Aeroclub, and Say Yes. Um so Wizard has a little video to pass to you guys. Now let's gonna watch.
Hello guys! Fiquem ligados, a Wizard, maior rede de idiomas de todos os tempos, com mais de 30 anos de mercado, já formou milhões de alunos em todo o mundo. Isso porque oferece uma metodologia exclusiva de ensino, com 2 modalidades por semana, em que o aluno aprende 100 frases por dia e é avaliado diariamente, na fala, audição, leitura e escrita. Nossa escola oferece 2 modalidades de ensino, que podem ser no formato presencial ou online, com horários flexíveis. Bom demais, não é mesmo? Tem mais! Nosso material didático consta de mochila ou pasta exclusiva, acompanhado do assistente pessoal, o SME, com diversos áudios e atividades extras, para que o aluno dê continuidade aos seus estudos, onde quer que ele esteja. Além disso, a Wizard oferece clubes de conversação e aulas extras, para que os alunos se acostumem às situações reais, monitorias e study hall, para garantia de aprendizagem do idioma. E mais, o aluno pode iniciar o seu curso a qualquer momento, porque a excelência da Wizard está primeiramente no atendimento de qualidade e respeito ao público. Portanto, inglês, para você falar, entender e nunca mais esquecer, só a Wizard tem. So, come to Wizard!
A Wizard, maior rede de idiomas do mundo, conta com uma metodologia revolucionária que tem por objetivo estimular a aprendizagem do aluno por meio do aprimoramento de todas as habilidades e o compromisso de não deixar ninguém para trás. Por isso, inglês para falar, entender e nunca mais esquecer só tem um nome: Wizard. Wizard, você bilíngue. Em Lavras, na rua Doutor João Silva Pena, 54 Centro. Telefone 3821-2601.
Thanks. Now we're gonna have the lecture. The Maria Fernanda Penaflor. She's biologist and professor in UFLA of entomology, and now she's gonna lecture for us. Just 1 second.
Good evening, everyone. Hope everybody's fine. Today, I'll be talking about sustainability challenges in entomology. And I will start presenting here my screen. So, today I'll be talking about entomology, but more specifically about insect chemical ecology. If you don't know this research line, you will learn along my lecture. Okay. So I am a professor here at UFLA, and I work in the Department of Entomology. And specifically, I work with a chemical ecology of plant-insect interactions. Okay, so you might know that several insects that are herbivores can become pests in agrosystems. So we have several groups of insects that are pests in monocultures. For example, thrips, beetles, bugs, caterpillars, spittle bugs, white flies, root worms, and aphids. All of them are insects and they become pests in agrosystems, causing damage to the crops. Okay. Well, we have a challenge, which is the climate change. Over the last decade, the mean global temperature increased by 1.1 degrees Celsius. And what about the insects? Well, it depends on the species, but insect pests in general, they will be affected in ways that increase their population. So temperature increase will likely increase the population of insect because of increased number of generations of insects, the expansion of geographic range, especially from the tropics to the temperate region. We will have more outbreaks of plant diseases transmitted by insects because insect populations will be higher. Well, increasing overwintering survival, so insects are more able to survive over the winter, which is not most serious and not severe winter, okay, among other effects. And also climate changes also includes changing precipitation, and this precipitation can be heavy or sometimes no rain, causing drought in agrosystems. And this will impact insects too. Heavy precipitation at the same time that can wash away the insects from the vegetation, they can also decrease overwintering. Again, survival of overwintering insects will be higher. So the generation after winter will be greater than the one from last year, for example. And the drought can impact insects because the plants which are under this stress, the water stress, they become more susceptible to insect herbivores. So they are more attracted to plants under water stress. So in this way, we expect that climate changes will increase the severity of pest infestations. Moreover, on the top of that, we have increase in a global human population, which is always increasing. We are already 8 billion people, and we have to produce food for these people. And well, will be another challenge associated with the climate conditions, which will cause more pest outbreaks. And today, the main tactic to suppress pest populations is by using chemical control. So chemical insecticides. Plants are treated in several ways, through spraying, aerial spraying, or manual spraying of the plants, or even with the drones, or through the treatment of seeds with insecticides. These insecticides will circulate into the phloem, and when the insect feed on the plant, they will get intoxicated. So another problem of using this tactic so much as we are using now is that pest populations with resistance to these insecticides tend to increase. So they get more resistant. This is a selection pressure to the pest population, selecting those that are able to detoxify from these insecticides. And when we apply a lot of these insecticides, what happens is that it impacts other insects. There are no target insects. For example, bees, which are so important for food production. So spraying insecticides unfortunately cause impact not only in the environment or also in no-target insects, which includes bees and diversity of insects, which are maybe natural enemies and in natural enemies of the pests, which these insects feed on the pests, for example, or insects that are important for the maintenance of life in Earth. And are there more sustainable practices to fight against insect pests? Of course there are. We have several, but how can we apply them? And we need to adopt several practices to suppress population of pests in ways to reduce the damage, the yield losses in crops. And for example, we have a biological control tactics that rely on natural enemies. Natural enemies, I mean by insects that feed on insects, especially herbivores. We have insects that parasite, for example, the insect herbivore. And once they are parasitized, these herbivores, they eat less and they eventually will die. We have also predators, insects. Predator, they eat, they not parasitize the insect, but they eat the whole insect. We have, for example, ladybugs that feed on aphids or feed on mealybugs. So we have other practices, of course. We have pheromone-based strategy. This deserves another lecture. It's another topic that I work on. Plant resistance, selectivity to natural enemies. There are so many practices to use to suppress pest populations. But today we're going to look into this system: plant, the insect pest, and its natural enemy. Okay? Well, to understand, first we need to introduce some concepts about plant-insect interactions. A serious threat to plants are the herbivore insects, which are potential pests in agro-ecosystem. And so plants, they are sessile organisms. They cannot move. They cannot run from insects. But of course, they are not like so susceptible to insect attack. They have, first, they have constitutive defenses. What are constitutive defenses? Are the defenses that plants have constitutively in their tissues, independent of, regardless of damage, insect damage. So it's the baseline of plant defenses. So insects will attack, but if plants didn't have this defense, they would attack even more and would threaten their survival. And another group of defenses is the herbivore-induced plant defenses. So these defenses, they are synthesized by the plant once the plant is attacked by herbivores. It's a way to save energy because they don't need this. Once they are not under attack, they can invest this energy in growth, reproduction. But once they are attacked by herbivores, they start to synthesize more defenses against these herbivores. So plants will allocate energy to synthesize herbivore-induced plant defenses. And when we are talking about this, we are talking about chemical defenses or secondary metabolites. So the plants have primary metabolites and they have secondary metabolites. The secondary metabolites will have a defensive function to the plant against insects, pathogens, and other stresses. Well, when we are talking about defenses against herbivores, then we talk about direct defenses. So direct defenses are defenses, chemical defenses, that will be detrimental to the biology of insects. They will like disrupt the development of the insect, or they will cause negative effects on the development and growth of the insects. Or they can be repellent. The plant becomes repellent to herbivores. So this will directly affect herbivores. And another type of defenses, plants can have indirect defenses. These defenses will not target directly the herbivore, but they will recruit natural enemies of the herbivores, the ones that I told you before about the biological control agents. So these are predators of herbivores or parasites of insect herbivores. And these defenses, they are in general are volatiles. These defenses are chemicals. They travel in the air, so they are volatiles. And these are chemical signals that the plant release, and that they will recruit. These signals will recruit the natural enemies of the herbivores. That's why it's called this defense indirect defenses, because they will control insect herbivores through the recruitment of their natural enemies. Okay, it's like a cry for help of the plant to control, to avoid the herbivore. Well, and when we are talking about crop plants, what is happening? It's a tough reality, because these plants, these hybrids or commercial cultivars, what is happening? These plants have a high productivity. They will provide bigger fruits, will grow fast. The plants are already selected for traits of productivity. So we want a plant that grows faster. The plant architecture, for example, it facilitates the management in the field, or the yield are bigger fruits, a great number of seeds. But at the same time, so this resource in the plant is allocated to produce growth and reproduction of the plant. So we can produce more, but at the same time, the plant is weak in defenses. So the insects are having a party in this hybrids and commercial cultivars, because they are so weak in defenses, in natural defenses. All the resources are being allocated to grow and to reproduce. But when we go back to domestication, when these plants were domesticated, so more wide genotypes of these plants were really strong. So the insects were, the plants have toxic compounds to insects. The plants were really strong, had strong defenses, but didn't produce as much as we want. So domestication is a process that made the plants weaker, but able to be cultivated. And then genetic breeding came and selected the best genotypes for grow management and reproduction, right? But these plants are really weak. And now, is it possible to go back and maybe having these wide genotypes and working with introducing traits of defenses against insects? Yes, we can. But we can also work with these cultivars that are already commercially available. Okay, so the answer here is Yes. And today I'm going to show a research from my lab that is have worked in a practice to improve plant defenses in crop systems. This, I introduce you biofertilizers. Biofertilizers are fertilizers that can increase plant productivity and are organic compounds or organic substances or microorganisms, for example. So we have inoculants of beneficial microorganisms or plant growth promoting bacteria. We have vermicomposts, which can be humic-like substances and extract from algae, amino acids, for example, or hydrolyzed proteins. These are all known biofertilizers for plants. But these biofertilizers can work as a biostimulant, right? Because the biostimulants here is because they promote enhanced nutritional efficiency, growth, and tolerance to abiotic stresses. But these biostimulants, we have the literature have been showing that these biostimulants not only promote tolerance, plant tolerance against abiotic stresses, but also to biotic stresses, because plants increase the concentration of existing defenses or existing secondary metabolites, or they increase the response to herbivory. They can synthesize quicker or more vigorously their defenses once the plant is attacked. And today I'll be talking about two biofertilizers or biostimulants, which are beneficial microorganisms and vermicomposts. Indeed, we are talking about humic substances in this talk today. Well, we published a review about the effects of beneficial microorganisms, which include plant growth promoting bacteria and humic substances or humic acid. Humic substances or humic acid, they are derived from organic matter. Okay, so it's totally organic. So in this review, we revised the literature and showed that a general pattern of the effect of plant growth promoting bacteria in the plant resistance to insects. And this pattern that we observed is that I will talk now about PGPB because it's easier. It's just the abbreviation for the abbreviation of plant growth promoting bacteria. So this PGPB, they increase plant defenses, direct defenses against insects. So we show in this review. And also the plant can defend more quickly and more vigorously once he's attacked by the herbivore. They can synthesize, the plant is in better conditions to synthesize defenses once he's attacked. And about the humic acid or humic substances, we found also that the fertilization of plants with the humic acid also increase the levels of direct defenses against insects. And this humic acid, it's the potentially mechanisms that the plant is more able to defend against insects is because of several processes that include hormonal modulation of the plant, the increase in enzymatic activity, and increase in the antioxidative metabolism. And this makes plants more able to respond defensively against insects once it's attacked. But the question that we raised in this review is that what about the indirect defenses? Indirect defenses are that defenses that recruit natural enemies of the herbivores. And what about the combined effects? If we just combine two biofertilizers, is there any potential of a synergistic effect in plant defenses? So we combine two biofertilizers, can we expect a greater effect, or their effect are not additive or synergistic? We expected that if we combine humic substances and the bacteria, the beneficial bacteria, we would have a synergistic effect, because when we applied humic substances, this promotes the growth of secondary roots and also radicular hairs in the roots. And this would be a surface to the bacteria, to the beneficial bacteria, to adhere and also to colonize the plant. So we would have a greater effect, a potential greater effect of these two combined, because then we would facilitate that the bacteria would colonize the plant. And the results I'll be showing here are from the PhD thesis of Lara Salles. She's already a doctor now, and she's employed, and it's very, very good to have grad students that are getting employed after grad school. Okay, so she studied the coffee system, and she studied specifically how can the two biofertilizers, so a bacteria here, a plant growth promoting bacteria, and the combination with the humic substances, what this causes, if this treatment, this application can enhance direct defenses against the white mealybug. Maybe you've already seen these insects. Sometimes people can make a confusion seeing this insect, because it seems like a fungus and a pathogen, maybe a fungus, but it is not. It's an insect. But they have a white powder on the top of it, and this can make people think that it's a fungus, but it's an insect. And we also here assess the plant defenses. What are the effects of the two biofertilizers and indirect defenses? Remember that when we revised the literature, we didn't see any effect about these biofertilizers in indirect defenses. And how this fertilization can enhance indirect defenses and recruit the natural enemies. So these were the two hypotheses that we had, that the biofertilizers would enhance direct and indirect coffee defenses against the mealybug, and that the combination of both biofertilizers would enhance even more indirect and indirect defenses. This organism here, is a ladybug that specifically feed on the mealybug. Okay, so this ladybug is not a predator of aphids, for example, but they are a specific predator of mealybugs. Here are pictures of the system. So we have here the mealybugs, white mealybug. You see the pounder, the white pounder over the insect. They produce lot of secretion, and this is the predator, the ladybug. It is not a common ladybug as you used to see, like the ladybug, which is like red with dark dots on it, but is a ladybug. And first we found before this study, we found that once the coffee plant is under attack of mealybugs, the plants start to release herbivore-induced plant volatiles that are really attractive to the ladybug, which is the predator of the mealybugs. So there's interaction between plant, insect herbivore, and the predator. And in Lara's thesis, she first investigated the biostimulant effect. Remember that the biostimulant effect is the plant, is the increase, the promotion of growth. And she saw here that the application of humic substances alone didn't cause a promotion, a growth promotion of secondary roots in relation to the control here, which was not fertilized by biostimulants. But once the plants were treated with the bacteria, then we saw a greater increase in secondary roots, and also the combination of the application of humic substances and bacteria. We saw a similar effect in the growth of secondary roots. And the shoot growth, we saw that all biofertilizers enhance the shoot growth. So it was heavier, the shoot. So the dry mass here of the shoot was greater than the control, but not different among the biofertilizers treatments. So we here conclude that there was a lack of synergistic effect on plant growth parameters. None of the combination of humic substances and bacteria didn't promote a greater effect on shoot growth, nor in root growth. And what about the direct defenses? How do fertilizers affected? How did biofertilizers affect plant direct defenses against insects? So we measured this in two assays, behavior assays with the mealybugs. We had this assay, which is called host selection. So we measure here the choice of the mealybugs, and they have here 4 plants, and one of the plants of each treatment. So we have control, plant fertilized by bacteria, plant fertilized by humic substances, and plant fertilized by both fertilizers. And we found that the plants that were fertilized by humic substances were the plants that were the least preferred plants. So they preferred to go to the control over all the treatments, the biofertilizer treatments, but they preferred the least the plants that were fertilized by humic substances. It was the least preferred, and even among the biofertilizers, okay? So biofertilization increased the plant resistance against the mealybug, but the humic substances alone was the greater effect. And another assay, the second assay to evaluate the plant direct defenses, was the biological performance of the white mealybugs. And we found here that in this assay, the mealybugs have no choice. So we just infest plants and cage these plants. So we involve the plant with the mealybugs with a cage, and we leave there for a while, for a couple of days. And then after this, we assess the number, we count the mealybugs, and we found that the fertilization with the humic substances or the humic substances in combination with the bacteria had suppressed the pest population, but the bacteria alone didn't cause any effect comparing to the control. So here only the treatments with the humic substances, either isolatedly or combined, presented deleterious effects to the mealybugs. And we assessed here as direct defenses secondary metabolite group, which is the phenolic compounds. So this is a proxy of the chemical defenses. And we saw that the fertilization increased the levels, the content of phenolic compounds in the plants. And before the infestation with the mealybug, and after the infestation with the mealybug, only these two treatments had these phenolic compounds increased, the bacteria and the bacteria plus the humic substances. And again, we saw a lack of synergistic effect of the combination of biofertilizers in direct plant defenses. Okay. What about the indirect defenses? So the indirect defenses here is the recruitment of the natural enemy, the ladybug, which preys on the mealybug. This assay is an interesting assay which we assess the olfactory response, the response to odors, to volatiles of the insect. This is called olfactometric assay. It's made of this olfactometer. It's a white tube. It's a Y-shaped tube. We release the insect in this main arm here, and we observe their choice if they prefer volatiles or odors, smell of plants infested by mealybugs and fertilized by humic substances and bacteria, for example, over plants that are infested by mealybugs but no fertilizer. And here is a video showing the behavior of this insect in assay like this. So introduce here the insect in the main arm of the UI tube. And we observe the behavior over 5 minute period. If they don't respond in 5 minutes, it's a no response insect. We hear in this white tube here, we have a flow, an air flow that passed through the plants and force the air through these two arms here. So there is an airflow in the system, and they have crossed when the insect crosses this line here. So we consider a response to the treatment. Well, in this assay, we have two treatments and we assess their choice, right? So we release the mealybugs and we see which treatment they prefer, right? So our first question was, do biofertilizers affect prey location by natural enemies through the emission of herbivore-induced plant volatiles? If we need to assess this response to herbivore-induced plant volatiles, we will infest cough plants with the mealybugs, right? Because then the plant will start to release this herbivore-induced plant volatiles. And we first evaluated the response of the ladybugs to herbivore-induced plant volatiles of no fertilized plants. So we saw that the predator preferred the volatiles emitted by plants infested by mealybugs over plants not infested. But when both treatments, undamaged plant and mealybug damaged plant, received the humic substances in the soil, we saw that the predator didn't discriminate between the two treatments. But when the plants were fertilized by bacteria, then the predator recognized the herbivore-induced plant volatiles, and the same happened here when we exposed the ladybugs to odors of plants, of mealybugs-infested plants, and no mealybug-infested plants, fertilized by both bacteria and humic substances. So this here is interesting because then if we fertilize with humic substances, then the predator may not locate their prey because they don't recognize that the plant is infested by herbivores here. So fertilization with the humic substance can hamper prey location by the predator. And when we asked if biofertilizers altered the attractiveness of herbivore-induced plant volatiles to the natural enemies, then we exposed the ladybug to volatiles of no fertilized plants over herbivore-induced plant volatiles of fertilized plants. And we saw that all treatments with the biofertilizers, they alone or they combined, the plants emitted more attractive volatiles to the ladybug over the plants that were attacked by the mealybug but were not fertilized. So we concluded from this assay that the biofertilizers enhance the attractiveness of mealybug-induced coffee volatiles to the predator. And here the last question in the olfactometric assays, we asked if there was a synergistic effect of the two biofertilizers on the attractiveness of herbivore-induced plant volatiles to the natural enemy. And here what we saw is that plants fertilized by humic substances release volatiles, herbivore-induced volatiles, that were as attractive as plants fertilized by both fertilizers, humic substances and bacteria. So probably the effect is from humic substances that increase. It's the same effect here of humic substances. And when we contrasted odors of plants fertilized by bacteria over plants fertilized by bacteria and humic substances, we saw that the ladybug preferred the odors of fertilization by both fertilizers over plants fertilized by only bacteria. So here, there was no synergistic effect in the combination of the biofertilizers, but a similar effect between humic substances alone or combined with GPR, which is the bacteria. So in this study, we found a bad effect here, which was the use of humic substances. They induce an attractive blend. So the fertilization with the humic substances make the plants more attractive to the ladybug, likely, because it was so attractive, the plant, an infested plant and damaged plant, as the plant was infested by the prey. And this is bad because this can hamper the location. The ladybugs, if they go to a plant that is not infested by their prey, they will have a deception, and they can learn that these signals, these odors, are not reliable of the presence of their prey. But we saw a good effect here of the biofertilizers, which was the combination of the bacteria with the humic substances. We saw that this treatment increased the attraction of the ladybug. We also assessed the hormonal modulation in the plant, and we saw here that the plant, no fertilized, but when the plant is infested by the mealybug, there is an increase of salicylic acid, which is a plant hormone important to modulate plant defenses against herbivores, but did not changed the levels of jasmonic acid when the plant was infested. We expected a greater effect in the hormone when the plant was biofertilizer, but we didn't see any significant effect. Indeed, here we saw suppressive effect of jasmonic acid in plants biofertilized by humic substances or plants that were inoculated with the bacteria. So no relevant effect. We found no relevant effect of the biofertilizers in hormonal modulation of herbivore-induced defenses. And well, to conclude here, we saw that fertilizers increased the level of phenolic compounds and decreased here parameters of the insects. So it likely increased the direct defenses because the colonization of mealybugs was less once the plant was biofertilizer, or in the multiplication was low. Some treatments with the biofertilizers. Regarding the indirect defenses, we found that the plant was more attractive. The plant released more attractive volatiles, herbivore-induced plant volatiles, to the predator of the mealybug. So biofertilizers, they have a potential to enhance the search of prey by natural enemies, with the exception to humic substances alone, which was a treatment that didn't yield good results. But we didn't find any synergistic effect, as we hypothesized first that the combined effect would, the combined application would have a synergistic effect. But still, more studies are needed to investigate the underlying mechanisms for enhanced chemical defenses. And to conclude here, the idea of sustainability. So in this study, we found that eco-friendly organic fertilizers applying to the soil, so which is a sustainable practice of fertilization, had a cascading effect, and it increased the plant resistance to pests and increased the recruitment of natural enemies of the pests. And this has a great potential for to increase the efficiency of biological control. So we have here the association of biofertilization and biological control through the recruitment of natural enemies from pests. Okay, but we need to look at this effect in the field and assess the efficiency of biological control to conclude that the potential effect in biological control. I would like to thank my group, Jessia in special, my student that invited me to give this lecture in this second webinar of Jessia. And I would like to thank all the organization for the reliance and the financial agency of this research. That is it. I thank you your attention here. And if you want more information, you can access the site of my lab, which is L'Equipe, the name, in Department of Entomology. And you can also follow in the social media ALAEC, which is the association, the Latin America Association of Chemical Ecology. And we have a meeting in the next month of this year about the chemical ecology, which is the science that study the chemicals involved in interactions. Okay, thank you. I'm open for questions.
Thank you so much, Professor. It was really, really great lecture. So now I'm gonna read for you the questions, if you
Okay.
think so. Okay.
Sure.
So Karina, that invited you, said thank you so much. It was great. And Emmeline asked, She said first, very interesting speaking. And she asked, what are the steps for PGPAB
Mhm.
application in the field? What factors must be considered for its success?
Wow. So bacteria, this PGPR, they're already available in the market. Both PGPR, I didn't mention here, but the PGPR and the humic substances, they are already available in the market. And there are several microorganisms that are sold by inoculants. And for their success, there are lots of factors to be considered. You have to consider the plant. So you cannot use bacteria from corn in coffee, for example. And so there is this specificity of the plant, right? And the association with the bacteria. Um, you also have the formula, the commercial formula, which is important to maintain these microorganisms alive to be inoculated in plants. Um, what else? Well, temperature, several factors, abiotic factors can affect the colonization of the plant with these microorganisms. And the bacteria that we work on was Enterobacter tabaci, which is not commercially available indeed. Was selected in another study as a potential PGPR, so plant growth promoting bacteria, correct? And well, there are lots of to do to make this available in the market. I'm not sure if I respond correctly, if she wants this.
Okay, she just said anything, we come back. So Julia said, really interesting and important study. And she asked, what do you think about transgenics? If you use it correctly using refuge areas next to the transgenic field, it can help on the case of using less chemicals, or it's also a planned response to a behavior.
Oh, transgenic. So I think that transgenic is a good technology because you reduce the amount of chemicals, the chemical control, right? So you can avoid spraying insecticides, and you can reduce the no target effect. For example, on predators, right? On other organisms that don't eat the plant. But the problem is the refuge, right? We know that that resistance is developed over time. So insects that are under great pressure of a transgenic BT technology, for example, they can develop quickly the resistance. But I think it's a good technology because the target effect is it's much better, and but don't get all the insects, right? So that's the problem. You still have to spray crops because the BT technology, they target few groups of insects, but the predators and the parasites that I told in my lecture, they are not targeted by BT technology.
Thank you so much. So now Yuki said hi. Thank you very much for the talk. The subject was very new and interesting, and I have never seen it before. And I had two doubts. Why healthy and product plant is so susceptible to pests?
Why
And the other,
healthy and
healthy and productive
they're so
is so
susceptible.
susceptible to pests.
Yes. So the problem is that the healthy plants, the hybrids and the commercial cultivars that we use it now, they have been selected for the traits, the characteristics of production. So growing faster in the field and producing greater fruits and greater number of seeds, and depends on the product, right? The organ, the plant organ we are interested in this crop. So we make grow faster, bigger, and so the energy, the enzymes and the carbon will be allocated to grow and reproduction. And this will have an effect on defense because they don't have enough energy to defend, but to grow and to reproduce. So that's the problem. Then the plant is weaker in defenses.
Great. And she asks, uh, and about these compounds that attract insects. Have you ever seen a case of this happening with products that are already been used in the crop management?
Could you repeat, please?
She asked about the compounds that attract the insects.
Mhm.
Have you ever seen a case of this happening with projects that are already been used in a crop management?
Well, compounds. So we can use this compounds from plants to attract natural enemies, for example. So we have it's a possible to buy these compounds and apply in crops. We have studies on that, and we can recruit more biological control agents to the crops by using some compounds that are available, that are synthetic versions of the volatiles that plants, for example, release. And we can take advantage of that to recruit more natural enemies. But the problem is we recruit the natural enemies, but we don't guide them to the infested plants. And then the natural enemy follows the cue, the synthetic compound, but they don't find the prey. Then they will learn that this compound is not a reliable signal of their prey. So it's a tactic that needs to be careful with.
Thanks. Now, Moisa said congrats. And are there regulations for the use of biostimulants?
Yes, well, in Brazil, it's there is two categories. No, indeed, is the category of biofertilizers. They are called biofertilizers, a biostimulant. It doesn't exist in Brazil. This category of product of, but there is the biofertilizer, and but I don't know about the legislation about this.
Okay. Now, Javier said, does it work with all biostimulants or with some species ones? Now we have a lot of biostimulants of different provenance.
Mhm. Yes. Well, I cannot generalize these results to other systems, especially bacteria. As I said, bacteria has a species specific effect. So a bacteria from found in corn, colonizing corn, I cannot use it in beans, for example. So we cannot generalize these results. And but we see a pattern that the biofertilizers enhance defenses against insects, but still the literature, they don't put this as a characteristic of biostimulant. They just mention about increasing tolerance against abiotic stress, but we see that the plant is more well defended against biotic stresses like insects.
Okay, thanks. Now Reinaldo said, good evening. Product wise, with the integration of biofertilizers as a more sustainable solution, how might they become more accessible and advantageous compared to the average fertilizers?
Gabriela, could you repeat the first term that you said? Because you said something with the bacteria, with the
Said product wise, with the
bread.
interaction of biofertilizer. I think you can read in the,
Oh, yes, in the chat.
yes.
Oh, here. Thank you. Product cut wise. Yeah, I don't understand this term exactly, product wise, but maybe it's well, we saw that the combination didn't yield a good result. So it's probably one biofertilizer that will enhance the plant defenses. It doesn't matter if we put another one in combination. And but I think yes, it's more will be more advantageous using these biofertilizers than the conventional fertilizers. But I mean, it's not. Oh, wait, it's a typo. No, yes. Well, but the chemical fertilizers they are more readily available to the plant. You can also like um, make specific fertilizers to the plant, like you can assess your soil is poor, in which nutrient the soil is poor, and then you can complement, you can add specific components that are the soil is poor. But the biofertilizers, no. But at least the humic substances, they are rich in phosphorus, and this is really good.
Thank you, guys. If anyone has any question.
Okay, thank you so much. Well, for students at UFLA, I have a course. I give a course in agronomy in biological sciences, which is called Feromônios e Aleloquímicos. I develop more about the subject, and you can check for enrollment in the next. Oh, there is another person here with a question. Sure. If there is time here with the organization.
Yes, okay.
Okay, sounds good.
Another one.
Yes, that previously used. Karina here. How long does it take to renovate or restore an area that previously used? I don't know, Julio. I don't know how long it takes. It's a complex question, very complex question. I don't have this expertise to know how much time. Oh, Andre here. How many repetitions are used with the photometer for you to be sure that there is a correlation with the insect behavior? Good question. So we use about 40 to 50, almost depends on the combination, because the chances of a random response is 50% for each arm, and so we need to change. You need to change the insect every time. Each replication is one insect, but we also need to change the source, the plants. So we change the plants and the insects. We develop a model to analyze these results. If you are interested, you can You can check with me in Department of Entomology if you are into statistics. If I am not mistaken, there was also another study relating Chrysopedius and bicho mineiro as a viable predator that also uses this method. Yes, there are some works being conducted with Chrysopedius and bicho mineiro in olfactometer assays. This is assay is very used. If it's if it's a study from UFLA, it's likely in my lab. Okay, I believe there is no
Yeah, I
doubt.
think that's it.
Okay, thank you so much for the interest, guys. It was a great time to be here. I hope to be collaborating with you in a in a future.
Thank you
Okay.
so much. It was so interesting. Thank
Thank you,
you.
Gabriela. Thank you, Karina, again for the invitation. Thank you, Jessia, for inviting me to this event. Sure, you can visit my lab. Sounds good. Bye bye, people.
Thank you. Bye.
Bye.
So, thank you guys. That's it for today. We are ending our second webinar, the first day. Don't forget, I already sent the first attempt list, and now I'm gonna send the second one. You need to answer both of them, and that's it. See you guys tomorrow with more interesting things.